Okay, so this is part 2 to my AP Bio phylogenetics videos. And so in this little short video, we're going to talk about how do you actually draw a phylogenetic tree? Well, or cladogram, right? So when we look at our sources of data, like what information do we use to draw trees? Well, we're going to use morphological, which is looking at different shapes or structures. So that could be like actual living species. We can study their characteristics and what they have in common, or it could be through the fossil record for the structure of fossils. Biochemical would be things like metabolic pathways that are present or absent in organisms. You have your molecular data, which your molecular data is going to be like DNA sequence or amino acid sequence, which really is going to be the most accurate and the most reliable data. So if given a choice, you're going to want to use DNA sequence or amino acid sequence, or the differences in amino acids or the differences in nucleotides between species to determine their evolutionary relatedness. Now, behavioral data can also be used as well. Now, I do want to point out when we talk about morphological data, why morphological data is something you should be careful of is because of convergent evolution. In a couple of my videos, I have talked about the marsupial animals in Australia and placental mammals in North America. Now, if they've evolved through convergent evolution to look very similar, if you're studying their fossils, you may not know just based on morphology how truly distantly related they are, right? So that is where more molecular data would be better, because convergent evolution can be a bit misleading on evolutionary relationships. Okay, so let's go ahead and see how we do this. So I have my 4 little made-up organisms that I drew, and what we see here is we're going to look at morphological data, and what we see is a character table. So the way we fill in a character table is I look at my organisms and I see that all 4 of the species have 6 legs. So in the character table, I'm going to put a plus sign for every species that has that trait. Now I notice in my species number 1, though, that species number 1 does not have blue spots, antenna, or wings. So for that, species 1 gets like a little line, like a minus symbol, right? But the other 3 species all have blue spots. Then I look at antenna. Species 1 does not have antenna. Species 2 does, but species 3 doesn't, and 4 does. Then I look at wings, and really that's only going to be in species 4. So here I have traits that are present in the different species and traits that are absent. So now we can use those characters or traits to draw the evolutionary relationship. So I'm going to start with like a cladogram style drawing. So here I see that all 4 species have 6 legs. So that is like a shared ancestral trait that was in the ancestor of all 4 species. But if I look at species number 1 doesn't have any other traits in common with the remaining 3. So therefore, after 6 legs evolved in an ancestor, there was a divergent event, right? Divergent evolution. So species 1 diverged from the other 3 species way far back in time, because there's nothing else in common with the rest of them. Then I look at blue spots. So the remaining 3 species all have blue spots. So on this line right here, this is like a shared evolutionary past. So blue spots arose in an ancestor to species 2, 3, and 4. It is a shared derived trait in those 3 species. However, when I look at species number 3, it doesn't have anything else in common with species 2 or 4. It really only has those 6 legs and blue spots. So there's a divergent a speciation event, basically, is what has happened here. Then we have antenna is next. That's in species 2 and 4. But then I noticed that species 4 is the only one with wings. Now, I just realized as I was talking that I didn't include this. Here I have traits that are all have all shown up in a species. Now, you could also have traits that are lost evolutionarily, like we see in snakes, for example. Like snakes and lizards do share a recent common ancestor. However, in snakes, their ancestor actually had legs, and then legs was selected against. So if you look at snake evolution, you can still see today the vestigial structures for a pelvic bone. So through the fossil record, we can see how limbs have been lost in snakes. And so it's not always gaining traits. You can also lose traits evolutionarily. Like there's in my video on evidence for evolution, I use the example of a blind cavefish. So there's a fish today who has an eye socket but doesn't have an eye. Like that would be a trait that has been lost, but its ancestor had eyes. Okay. And now some people prefer to draw, or sometimes you see the phylogenetic trees drawn in this style. So this would be how you draw it in this like shape or style, a phylogenetic tree. So they both show the same thing. Now, I do want to point out though, these highlighted yellow parts, these traits represent shared derived traits, which basically indicate common ancestry, and it helps you draw your hypothesis of how these species evolved throughout time. Remember, these trees are not just, but they are a hypothesis or hypotheses, and they are like able to be reconstructed or reevaluated as we gather more evidence. Maybe new fossils are discovered or new technology is developed, and we're able to refine these hypotheses and redraw them. That is totally possible. Science is wonderful because it can change as we deepen our understanding. Now, I also want to point out, though, this species number 1. We notice it doesn't have much in common with the others, so we call this species number 1 an outgroup. An outgroup is a lineage that is the least closely related to the remainder of the organisms in the phylogenetic tree or the cladogram. So 1 would be our outgroup, and then 3, 2, and 4 would be what we call like an in-group, and they have like shared derived characters, and we can see how their evolutionary path, basically. Okay, and so our last thing to look at in this video is how do we use molecular data, like I mentioned earlier, is one of the best sources of evidence or data to use when constructing a tree or cladogram. So in molecular data, that can be either, like in this example here, I have the number of amino acid differences. Now, if we remember, our DNA sequence is what codes for codons, which then codes for amino acids, and then that forms a polypeptide. So here we have protein X that's made of however many amino acids. And if we have like a missense mutation in an exon, that'll change the amino acid, but the protein may still be built. So that is what we're looking at. Now, if it's a silent mutation, then we really wouldn't see any amino acid differences. You'd only be able to tell silent mutations if you were looking at the actual DNA sequence. Okay, so let's go ahead and see how we fill in a table like this, though. So species A, we're going to compare the two species. So we're looking at like the number of differences between species A and species B, between species A and species C, etc. So if I were to compare the number of amino acid differences in protein X between species A and species A, you would expect to not find any differences because they're the same species. So in a chart like this or a table like this, we'd put just like a dashed line or like a 0. Like there's no differences because they're the same species. However, if I wanted to compare species A to species B, I see here there's 4 amino acid differences in their protein X. Now, if I want to compare species A to C, there's 2 differences. If I compare species A to D, there's 6, and A to E, there's 10 differences. So if I look at just this row, to me, what I interpret is that species A and C have a recent common ancestor. There's very little differences between them, whereas species A and E are more distantly related. Their common ancestor is further back in time. They've been separated for longer because we have these things called molecular clocks. And molecular clocks are basically like the rate of mutations. The rate of mutation is pretty constant across species, so you can kind of gauge how long 2 species have been separated evolutionarily based on the number of mutations that have shown up in their two lineages. Okay, now let's go ahead and compare species B to B. In this, oh, I'm sorry. Here, if I compare B to A, we already did that. It was 4. So we don't do it again. We can either do a dashed line or we can just shade it in or leave it blank. And now for B to B, again, we're going to expect no differences. And then we fill in the rest. Ooh, that one right there is pretty important. You always want to look for the numbers that are the smallest, which implies very closely related, a very recent common ancestor, as well as the numbers that are the most different. That also is going to tell you about your out group. Now, species A and C, we know had 2 differences. Species C and B, we know we had 6 differences. So now we're at C to C, and it's like they're the same species. Then we have 5 and 9, and then we continue filling in this table. Now, what do we do with this table though, right? Like, how do we use this to draw our trees? So let's go ahead and check that out. So here I have the table, and what I like to do is I like to start with the ones that are most closely related. So what I see here, oh, I also look for my outgroup. Which one is the most different is also very important. So here I have E as my outgroup, and then I notice that B and D have only one difference. So they have a very recent common ancestor. Then A and C also have a very recent common ancestor. And if you're looking at this, you might be thinking, well, that's not a cladogram or a phylogenetic tree. Well, for me, I find it really easy then just to connect the lines basically. So now I have a tree, and if you check the numbers and data, it really does work out. Now we also want to point out that these trees can be switched at the nodes. It would mean the same thing. Now we can also draw our tree this way. Oh, I'm sorry. So we can switch it at the nodes. All right, means the same thing. Now, if I look at the goalpost style, here B and D had only one amino acid difference, so they have a pretty recent common ancestor. And then we had A and C. They had 2 differences, so I drew their line, their more recent common ancestor, a little bit further back in time. If you remember from my last video, phylogenetic trees are kind of calibrated based on molecular clocks, like the rate of mutations and how many differences there are. Now, E was our outgroup, so that's a further back. So this is how I would draw this table in a phylogenetic tree style. All right, so that's it. I think at this point, if you're in a biology class, I'm guessing your teacher's next step is to now practice with different character tables and molecular data tables for you to draw your own trees. All right, great job.